Are we?
Yes.
Where are we?
Here.
Why are we here? Not entirely clear. We are misfits thrust into existence by random chance, with no hints at all as to how we're supposed to make sense of it all. It's immensely bizarre. Here we are.
Hello, everybody, and welcome to the Here We Are podcast. I am comedian and science enthusiast Shane Mauss. My guest today is marine biologist and science communicator Skylar Bayer is joining me. Skylar, why don't you give yourself a proper, more thorough introduction? Tell us all about yourself. What's your life story? Just set up a little bit of what you do, because I know that you're into I know that you're a marine biologist, but you do lots and lots of science communication. So if you could tell us a little bit about that.
Yeah, sure thing. Thanks, Shane, for having me on the show. And I don't know that your show is long enough for my whole life story, but right. I'll give you the highlights so far. Let's see. On the science side of things, I am an assistant professor at Roger Williams University in the biology department, and I do aquaculture and extension work as part of my job. Extension means engaging with the community, specifically around like aquaculture and fisheries work. So that's some of what I do, and this is my first semester starting. So it is a wild time to start a job as a professor.
That's a horror story that you just shared with me. That is the absolute worst possible situation to be in. How's it going so far?
I mean, it's, I will say that I am super, super lucky that I got this job. I signed my contract like right before the pandemic really took off. And there are lots of people that have had early career folks who are super vulnerable professionally, who've had negotiations just disappear as universities pull in their budget strings. And so I'm really lucky, and I work in a really great department. And I have to say, I'm a high risk person for COVID. I will get into that later probably. But so I teach remotely. I'm not on campus this semester, which is too bad.
They're doing in person. Where are you? Where's Roger Williams?
It's in Bristol, Rhode Island, and actually what they're doing is they test all the students and staff and faculty that are on campus twice a week. And they have changed so much on campus to make, they do social distancing, they do mask wearing, any student can access a class remotely. They've been doing a really good job of really trying to make the in-person part possible, but the testing twice a week is really the big thing that makes that work. And I think they've had less than 1% of the testing population test positive since mid-August.
That's amazing.
So that's actually pretty good considering some of the horror stories that have happened on other campuses. And they've contracted out the testing through this institute called the Broad Institute, which is based in Massachusetts. And there are a lot of small colleges throughout New England that have contracted with them to test their student population regularly. There are different schedules for different schools and what they think they need. So, people are making it work for the most part.
What are the results? I mean, how long does it take to get a result with their tests?
I think it's like 12 hours at most or something like
bad.
that. It's like next day kind of results is my understanding. That's just what I've heard. I am not in the testing program because I'm remote this semester, but yeah, I think that people are mostly happy with how it's going, although, there are bumps in the road. But they're figuring it out as they go, right? There's a lot of building the plane while flying at situations happening this year, right?
Yeah.
So, and that includes all universities, right? So. Yeah.
I've sat in several classes of different past guests and stuff this year, and it's been interesting to see how different people are managing. I will say I'm in outside of a little city called La Crosse, Wisconsin. It's not that small. It's like 40,000 or something like that. and I'm a small town outside of it, but I've had two scares from in my like bubble recently. Like, you know, someone in my bubble had been in contact with someone that was positive. It happened like twice. It hadn't happened at all. It happened like twice in 2 weeks. And then we went to look into testing, and here they won't even let you get a test if you don't have symptoms, which is insanity. And then which is like the whole problem with COVID is the asymptomatic spread.
Right.
If we were just relying on symptoms, there'd be nothing to worry about. I mean, not nothing, but it would be such a smaller threat than it is. But then you can go, like, there's certain things like once a week or every other week, or like some city nearby you can drive to and pay for, and like all these, you know, there's ways around it. It's not easy, and those results are like 4 to 5 days.
Wow.
Which is like,
it's a long time to wait.
That's a really long time to wait. And all our bars are open here and everything else. It is a tricky situation. But yeah, well, that's interesting to hear, that's the way things are going on over there. It sounds like it's pretty sensible.
I wouldn't say, I'm not that envious of everyone that's had to be on campus. I think it's stressful teaching remotely, but it's a different kind of stressful having to deal with all the logistics on campus. And although I've just joined the department, I've been really impressed with my colleagues, both in the staff and the faculty, who've made a lot of things work and have been a huge part of making the COVID testing happen. Even, you know, like they're the ones that really helped figure out how the university could contract out to the Broad Institute to do twice-a-week testing. So, you know, I think that everyone is dealing with a lot with this, but I've been pretty impressed so far as a new person on campus.
Yeah, amazing. Well, let's get into your work specific. First off, what is the science communication stuff that you do?
Yeah, sure.
You have your own...
Well, let's see. So I guess I'll back up. When I was a grad student, I got really interested in science communication for a couple of reasons. So I kind of dabbled in a lot of stuff. I had my own podcast for a long time called the Strictly Fish Wrap Science Radio Hour. It was actually out of a local station where the signal didn't even really leave the town in Maine. And then I did a bunch of
in Maine too.
Yeah.
Why didn't you just put it out digitally? There's no one in Maine.
I did.
Oh, okay.
I did.
Okay.
Yeah, I lived in Maine for almost a decade.
I love Maine.
Yeah,
where in Maine?
It's really great. Mid-coast Maine. How well do you know Maine?
Well, I lived in Boston for 6 years, and
So you know where Acadia is and Portland is.
Yeah, I mean,
Yeah.
I did shows all around New England. Didn't get to Maine that often, but lovely.
Yeah, it's really a great place. And so, and I worked on a fishery species for my PhD, the sea scallop. It's a big federal fishery, and then it's also an important state fishery in Maine. I think Maine is the only state that has a state fishery for sea scallops. So a lot of my work involved communicating with fishermen and management folks, and so I had a lot of interest in communication. So, and then I got involved with the Story Collider, which are you familiar with them, Shane?
That's why we were put in touch originally. That was so many. What was that, like 2 months ago that we? I kind of forgot. Yeah, Story Collider. Tell the audience all about it.
Yeah. So if you're familiar with programs like The Moth, like the personal on-stage storytelling, usually their stories involve something happening that's meaningful in their life. And so Story Collider does something similar, not different, different in that almost all the stories have something related to science in them. So it might be a scientist telling a story, but it might also be someone who's had a medical experience, right? Which is a lot of people. Or they took a really interesting class in high school, or, there's a million different potential stories out there. And so I got involved with Story Collider in 2013 when I told a story for them about going down in 𝕊𝕐𝕦𝕒𝕟, the submarine, because I used to do deep sea work, and how I got to call my dad from the bottom of the ocean. And that was a particularly important story for me at the time because my dad had just had a really big stroke. And it was a hard time, but I had this outlet at Story Collider to tell this story. And then I asked them, I said, well, can you guys come to Maine and do shows? And they're like, yeah, and you could be a co-producer. And I was like, I've never produced a show. I'm a grad student. And then I don't know, I started helping producing shows and I really liked it. And I've been involved with them here and there as a producer. I don't produce as much as some of the other producers, but I've been producing shows for them on and off for about, I think, 6 years. And I'm friends with a lot of the other producers. It's a great group of people.
I was just talking about this recently where I was explaining, I blab a lot about my personal life and I, make myself very vulnerable and stuff and it's not even that I don't dislike it. I just don't care one way or another. Like I'm an open person. I don't like whatever. I don't get like a rush out of being vulnerable or anything. But the reason why I do it is because most people connect so much more. Like I love talking about the big concepts. I love getting into like the nitty gritty. I like being like an objective observer too. And I don't really care about like my personal stake in things as much. I like stepping back from, but boy, people sure attach to that personal narrative. And I think it's really important in science communication. We're storytelling animals, as you know, and it's just it's just such an easier tool. That and graphs. I mean, people love graphs too. I don't know what that has to do with evolution. We definitely evolved to be storytellers. Why we love graphs so darn much? Maybe they've studied that, but
well, I think a lot of people are visual creatures, I guess. Not everyone, but a lot of people are. So that would be my guess about that. But I'm not a psychologist or a neurologist, so I'm not sure.
It also seems very sciencey. Like when you see a graph, there's something about it that makes people be like, well, okay,
that's legitimate.
This person has a graph. They drew 2 circles, labeled them. I think they know what they're talking about. Right. Yeah. See, it gives it legitimacy if you make a figure and make it look pretty. Yeah. Yeah. Do you have you so this is your first year teaching? I'm wondering if you've tried to take any of those aspects from what you've learned from your science communication and Story Collider and such in your style of teaching. Yeah, totally. And I've used it for a lot of things, and there's a lot of projects and things I've gotten involved with because of Story Collider and the people I've worked with through Story Collider. So this is going to loop back to the teaching in a second, but one of the things I did is I took a Wikipedia editing course because through 500 Women Scientists, are you familiar with that group? Yes. Yeah, it's like an activist group, mostly women. I think it's all women, and or female identifying folks. And one of the people who's in the leadership group is a friend of mine, 𝕊𝕙𝕒𝕦𝕤𝕠𝕟, who's also another producer for Story Collider. That's how I met her.
And she and this woman, Jess Wade, who's a physicist, have worked really hard on getting more women scientist biographies on Wikipedia, because there's a real disparity between men's and women's, like the amount of biographies of a male and female scientists. And so they offered this Wikipedia editing course because there's also a dearth of female editors on Wikipedia. Like it's like something like less than 15% identify as women as editors, and it's a volunteer community. So I applied for this course, and I did it back in May, and it was a lot of fun and very empowering to feel like, oh, now I understand how to edit things on Wikipedia, and it's great because it's this free open access resource. You learn how to cite things properly, what are good resources, all that kind of stuff, and it helps improve a very accessible resource to people to get the right information. And so then I found out you could apply to do a Wikipedia course with your students, like do a Wikipedia project. So the final assignment for one of my classes, actually the class I had before this interview, is a Wikipedia assignment. And so the Wiki Education Foundation does this great training program, and there are all these modules and exercises they prep so you can become comfortable with it before you just start putting in text on Wikipedia. So that's one way I've incorporated what I've learned. And then also I tend to use GIFs a lot in my slides. Nice. And I'm very honest with them. This is a rough
stories,
semester.
graphs, and GIFs.
Yep. Yes. And I tend to try to pull from current news articles, things that are fun. Like today I found a Halloween fish article I shared, like the scary fish we eat from the deep sea. And then another thing I do is I've started assigning some podcast assignments. There's been some work done that shows, it's called Scientist Spotlights, or Spotlight. I don't know, if you Googled Scientist Spotlight articles or something, there's some literature that's been published that shows that when you have students listen to either podcasts or read articles about scientists, and like a diversity of scientists, like demographically, not just the same boring old white guy in a lab coat kind of profile. Learn about
need more exciting old white guys in lab
right.
coats. Is that what the issue is?
Perhaps some women or people who aren't white, people of color.
What?
Yeah, right. Well, and a lot of students don't necessarily know that if they're not an old white dude, that they can be a scientist too, right? Because we tend to want to see examples of ourselves in groups of people. Like, oh, I like science, but I don't see anyone like me there. Maybe I don't belong there, right? Mm-hmm. So doing these exercises in class where you learn about the scientist and their work, you learn about a lot of different kinds of scientists from their background or their research, and weave that into the science you're learning. They found that it makes students feel like they belong more in science, that it's more accessible, they're more interested in it. So adding that personal story dimension to learning science, right, just keeps people more engaged and feels like it's something they belong to more. And so I'm hoping to do more of that through my classes, as I get adjusted to teaching the courses I'm assigned to teach as well. So,
yeah, that's amazing. I it's actually a big part of why I grew out all of this quarantine hair, is to inspire the homeless to get more interested in science themselves, to be someone that they can look up to. Oh
man, your beard reminds me of a Portlandia episode I just watched.
I've never seen Portlandia, and I know that wasn't a compliment.
Well, so they have a whole episode or clip on how kids aren't exposed to enough germs anymore because we keep them so sterile in cities, and so they can get beard dolls that are grown from men with beards and then harvested, and their beards are exposed to all sorts of germs.
Yeah, that is a genuine concern with this with masks and everything else. Mm-hmm. But I've been exceptionally cautious and just distance extra. I do like 8 to 10 feet instead of 6 because I throw on a couple more feet for the beard.
Right.
For the added
a foot per inch, right? Or something like that.
Yeah. But yeah, I definitely think that it's as a science communicator myself, as someone that that works hard to have a diverse array of guests. It's not the easiest thing to do always in academia. And I know it's easier now that I'm doing things remotely, and I should really I've been meaning to kind of up my game a little bit in that regard, but touring around with a live show where I'd have 2 scientists join me in each city. Me and my assistant as well would be, often that was one of the things that we really had an eye out for, was any time that we could include more diversity in the shows. And I think it's a lot better than it maybe was 2-3 decades ago, but I think that we definitely have a long ways to go.
Yeah. And so since we're still talking about diversity, I should tell you that there's a book project that I'm working on with my collaborator, Gabby Serrato-Marks. She just got her PhD, actually. And we both told stories for the Story Collider. Well, I've told a bunch of stories for the Story Collider, but we both told stories about dealing with medical condition in my case and a disability in her case in field work. I have a heart condition that at the beginning of my PhD rendered me unable to do scuba diving anymore. And so, but I worked in a scuba diving lab for my PhD. I'd already been admitted and all that stuff. So I spent a lot of time on the boat alone in Maine on the coast waiting for divers to come up. And that was,
you just broke my heart. I'm so sorry.
There's far worse things.
Yeah.
But the artistic director at Story Collider, Erin Barker, who if you haven't met, she's like one of my favorite people in the entire universe. She and I have like a mutual fan club going on. But she suggested that Gabby and I get together. She's like, you guys should, I don't know, maybe write a book or do a project or something. And so what we did is we solicited a lot of pitches from scientists with disabilities or medical conditions to share their personal stories of what it's like, being a scientist with their conditions. They're not all sad stories. They're like happy stories. There are certain advantages or skills you develop when you're kind of different in your field. And we ended up, we're under contract with Columbia University Press for our anthology book of all these stories called Uncharted. And so the book is due to the publisher next July, I think. So hopefully it'll be out in a year or so, but we're really, really excited. And it's honestly one of the most fun projects I've ever gotten to do. And I've gotten to meet a lot of really cool scientists. So.
Well, that's amazing. That's very inspiring. Actually, hold on one second. I'm going to show you something. This is different, but related. I take the covers off all of my books, but there's this, I haven't even finished it ever, but there's this Injured Brains of Medical Minds.
Oh, cool.
You might want to check it out. It's all people that like spent their entire career studying like Alzheimer's or whatever, and then got Alzheimer's, and then are able to like articulate the experience from the point of view of an expert, or they studied bipolar or what, and then have a manic episode themselves. But anyway, thought you might be interested.
Yeah, definitely. All that stuff is really cool, because one of the things that I think people struggle with is like normalizing having medical conditions or disabilities, and all of us usually end up having some sort of condition at some point in our life, and it's okay to be different and try to figure out ways to work around certain issues or work with issues usually.
Yeah.
And I think in science, there's a lot of expectations of working yourself to death. And if you can't do this, then, you'll never be useful in this field kind of stuff. And it's not true. There's lots of ways to contribute and have unique ideas. And Gabby and I actually wrote a piece in Scientific American last year about some of the advantages we've had having conditions to work with and the positive side of it. So
very cool.
Yeah.
Like what? Give me a for instance.
Well, so because of, I'll talk about myself because I'm most comfortable talking about my case. But I have this heart condition, and I actually have an ICD implanted in my chest. I can feel it. It's right here. And that got implanted almost 9.
How long did that take to get used to? That you can feel a thing.
I think it took a couple months to get used to. I was 25 when it happened. And so the full story is that when I was born, I had heart surgery. I had what's called the transposition of the great arteries. And so that means
that sounds amazing. It sounds like a novel or like a great novel or something.
It's like an epic, tale.
Yeah. That
would just be the title of my life story, right? The transposition of Skylar Bayer's great arteries. So what that means, though, is that, your heart kind of does this figure 8 circulation of blood, right? The veins bring in the deoxygenated blood, then they get pumped to your lungs, which is an artery, right? That's a major artery. And then the lungs oxygenate the blood, they bring it back to the heart, and then the heart pumps it into the aorta, which goes to the whole body. And so it's sort of this figure 8 kind of system. And so what happens with transposition of the great arteries is you just sort of end up with 2 loops that are being pumped by the heart, but they're not connecting. And so that's fine when you're in utero because you don't need to breathe. But then as soon as you start needing your lungs, that loop is still cut off from the rest of your body. And so unless you have surgery, you usually end up dying eventually. Wow. So I had that, and I didn't really have any problems most of my life. I'm considered very accomplished for a cardiac patient, I think, by my doctors. And so, but when I was 25, I got really sick and started having these weird little arrhythmias. And it turned out that whatever I was sick with sort of uncovered this condition called polymorphic arrhythmia, which just means many-formed arrhythmia, which doesn't really mean anything. It just means the electricity in my heart is kind of messed up, I guess. And so they couldn't fix it, basically. They couldn't figure out why I was having these arrhythmias. And so they just put in an ICD, which is like an intercardiac defibrillator or cardioverter defibrillator. And so it's there to defibrillate me in case I need to be, or it paces me. It doesn't pace me, though, and I've only been defibrillated once by it. And so, let's see, that was 9 years ago. And so then that happened right at the beginning of my PhD. And so I couldn't scuba dive, but I'd been scuba diving one season. So I kind of knew what the bottom looked like and what I was supposed to do. So I became a really good project manager, which is a really important skill for a PhD anyway. So I knew how to tell people what to do when they were diving. I wasn't tired all the time from scuba diving, so I had more energy and time to plan things properly, to make sure I had time to do my other experiments and coordinate with everyone. And I probably got a lot more work done overall than if I'd been scuba diving, is the short version of that. So project management and leadership skills are really critical if you're going to be running a lab or anything like that.
And I think the takeaway from that whole story is scuba diving is a big waste of time. Is that what you're going?
It's fun. It's fun.
I would love to go. I can't believe I've never been scuba diving. All of the adventures I've been on in life, and I've never been scuba diving.
Really?
It's crazy.
Yeah.
I'm on the move too much, I think. Maybe I need to like be in a place for a while. I also have a subtitle for your book, which is How I Fixed the Electricity in My Heart.
Yeah. No, that would be pretty good.
Throwing it out there. We can workshop it.
We'll workshop it later, Shane. Yeah.
So tell us a little bit about some of your marine biology background then.
Yeah, sure. So let's see where to start. I guess
considering we're halfway into this episode, maybe we could. I suppose we should start talking about science. Eventually.
Right. Well, the communication part is fun.
Yeah.
So, I got my start doing deep sea work actually, and started an internship in an undergrad, and then I ended up doing my master's in the lab at Woods Hole Oceanographic Institution. And I got to study deep sea hydrothermal vents. And are you familiar with those, Shane?
Kind of. I had someone on just before COVID that was a volcanologist. Am I pronouncing that correctly?
Yes. Indeed, you
spent
are.
a lot of time in a submarine, and told me the most amazing stories about it. And so I know a little bit from that, and having seen them in like David Attenborough documentaries and stuff.
Right.
Let's just assume that I've never heard of such a thing before.
All right. So, and if anyone's watching this, you should just like Google hydrothermal vents because the pictures are amazing. But, the Earth has plates, right, of crust that moving back and forth. And then they make volcanoes and eruptions and earthquakes and all that stuff. And it happens on the surface in some places, like, say, San Andreas Fault or other volcanic regions, which are escaping me at the moment. But there's a lot underwater too. And so there are places where there's volcanic eruptions underwater. And what happens at these places is that the very, very cold, dense salt water seeps into the cracks, and it gets heated up by the magma underneath and then comes shooting out in these vents, which have lots of, what to us anyway, very toxic compounds, like sulfur and heavy metals and all sorts of stuff. But there are all these animals that only live at these hydrothermal vents. It's really, really amazing. They can only be found at these hydrothermal vents, and a lot of them either eat or have incorporated in their body bacteria that specifically digest those super toxic compounds, especially the sulfur ones. And so the whole ecosystem is based on those super toxic compounds coming out, and the fact that it's warm. I think that helps as well, right near these vents. And so there's like limpets, these little snails and crabs and giant tube worms in some cases. And there's actually quite a lot of diversity, depending where you are in the world, of organisms. And I was studying for my master's this one area. It's called 9 North on the East Pacific Rise, which is kind of near Mexico. So you fly to Mexico and take a boat out to the middle of nowhere. And 9 degrees north is 9 degrees north of the equator. So it's very warm, tropical area. And what happened is that there was an eruption, I want to say like 15 years ago. And what scientists wanted to do after the eruption is study how the community of animals changed over time. It's called succession. And it was a rare opportunity because usually you don't get to know when those eruptions happen. But I think like a geologist or someone had a seismic device that stopped beeping because it got covered in lava. And so they're like, oh, this happened. Let's take the opportunity to study these animals. And so I spent a lot of time looking at animals under a microscope, scraping sulfur off of little plates, and then cutting them up to find out how they reproduced. So that was pretty cool. But the problem with deep sea work is that you have to have big grants to go on big boats, and you don't get to go very often. And so I really liked one cruise I got to go in 𝕊𝕐𝕦𝕤𝕪, the submarine, and go down to the bottom and see those animals and stuff. It's really cool. But then the lab that I was in basically didn't have money anytime soon, and I needed more projects or whatever for if I was going to stay and do a PhD. So I left with a master's and I went to Maine instead to study sea scallops.
Can we, before we get into sea scallops?
Yes, I figured you'd have a question.
I have at least two pretty big ones.
Yeah.
Maybe this first one, I don't know if this first one's a good question or not. The second one, I think we're going to sink our teeth into. First one, drum roll, please. I was thinking, I didn't realize, speaking of David Attenborough, I was just watching some documentary or one of his newer docuseries, and they were in South America and talking about the volcanic activity there there was something, it was like some of the volcanoes erupt with the force of like 100 or 1,000 atomic bombs or something like that. I wish I could remember the statistic up, but I didn't realize that as you were talking, I didn't realize that these vents were something that stayed so active for long enough to create like a evolutionarily stable system where you have
like an ecosystem.
A whole ecosystem where species are able to evolve and adapt to that in enough time that the vent's still there and active and be able to make use of it and sustain. And then what happens when these vents go away? Do like just entire species just go extinct once?
Well, so it's a really good question, and it was actually like the focus of a lot of the researchers I worked with at the time. And so one of the ways that these organisms persisted is that they put their larvae, their babies, in the water column, and they must do it. I think a lot of them, that was one of the things we were sort of trying to look at, is if there were seasonal patterns or not. And the expectation is there wouldn't necessarily be a seasonal pattern because you're not dealing necessarily with food that's created at the surface from sunlight. But also, I don't think it would be advantageous to only reproduce one time a year because you don't know when a volcanic eruption is going to be. So it's good to always have some babies in the water so they can recolonize a new area. But yeah, the volcano will come and wipe everything out. And that's one type of like deep sea community. There are other deep sea communities that live off of like methane seeps, and those communities are much more stable. And so it's possible that some species evolved in one system, and then eventually some colonized a hydrothermal vent and evolved there too. So it's pretty wild. It's wild that it's possible.
It is. I love that so many like fish and things do this like crazy lottery. I mean, at the end of the day, I guess it's all a pretty crazy lottery system that we're in. But just to like the idea of just like having thousands of eggs or whatever all at once,
or millions.
Oh, millions. Or well, I guess we'll see what happens. Maybe some, not to overly anthropomorphize too much, but there is something like deeply it just like rings true for like it's symbolic of the universe or something, that these little creatures are just like, I guess we're just gonna squirt our stuff into the abyss and cross our fingers that and life so comes of that.
What's funny about that too is like most sea creatures, I've tried to find a number on this and I haven't had much luck in recent years, most marine organisms do, like if you look at species, they do that kind of reproduction where, you know, it's like throwing confetti into into the water. And that was a visual that I used for my students actually recently when we were covering larval dispersal. And so, you know, some organisms will...
Those kids made a lot of jokes about that after
I hope so.
class.
I hope so. That's the point. Then they get to... Then they get to remember it. That's what I tell people. I make a joke out of it, and then I remember it. But on top of the larvae, right, there's what's called broadcast spawning, which is directly relevant to my next research chapter of my life, where they put the organisms put sperm and egg into the water column, and it's just like they'll fertilize, right, and then we'll get babies. And so it's another level of just like throwing it out there and seeing what happens, as opposed to growing, you know, larvae inside their bodies and then releasing them.
Right. What does that look like? First off, what exactly is a water column? Like, what?
It's just the water. It's just the column of water sitting on top of the bottom of the ocean, let's say. So it's called a column because we tend to think of it vertically, right? Like it's when we're thinking about where things are in the water, it's like, where on this column is it? I mean, I agree, it's kind of a weird name, but I think it comes from...
I'm glad I asked.
Yeah. I think it comes from the idea that a lot of the time in oceanography, we look at vertical profiles, right? And we're like looking at, this column of water.
I see.
Or like a core, sediment core, right? It's usually a column, and you're looking at where it is on the column.
Well, I'm glad I exposed my ignorance, because I was picturing like the thermal columns or whatever like hang gliders use to elevate, and that's what I thought they were. I thought these little fish were blowing their babies into these little drifts or something, but it's just...
Well, yeah, I mean, so I'm talking more generally, but I think one of the questions that researchers have looked at with hydrothermal vents too is, so those hydrothermal vents create a plume that sort of becomes neutrally buoyant at a certain depth, right? Like it rises, and then there's some depth at which it stops rising. And so there's questions about, do hydrothermal vent larvae get entrained, you know, in there? Is that how they spread? Do they go somewhere else in the water column? And I don't know the answer to that question, if it's something they've really figured out. I'm sure it depends on the species.
So is there, I mean, are they factoring in, is there some sort of sensory input they're using where like, oh, this is the condition that I want to, it's date night, I'm going over here, and then this is where I release my confetti. Is this, or do they just, or are they just like, oh, I have enough confetti stored up again to blast one off and then just get to work on the next?
Well, I think that the answer to that is it depends.
Yeah.
And it's I mean, what you're talking about is spawning behavior.
Yeah.
Without knowing
it would be amazing if I just listed the two only known spawning behaviors that there are. Scientists have thought long and hard for many years, and what they've come up with is sometimes they blow their confetti randomly, and sometimes they're looking for a perfect spot to blow their confetti.
Yeah, well, it's, you know, that might not be too far off.
That is.
And broad turns, yes.
That's ridiculous that I was even close to being anywhere near accurate with any of that.
I think so I'll tell you, there are a lot of spawning behaviors where organisms specifically group together to make sure a high percentage of the eggs get fertilized. So if fish can swim, they can aggregate into big clusters and make the distance between their eggs and sperm in the water column shorter, right? So you're more likely to get fertilization, and organisms like scallops or sea urchins or something that's on the bottom, they can move closer to each other. Then like you have coral reef systems where multiple species will all spawn on like the same night. So it's like timing is a big thing.
Yeah, I've talked with some coral people that got to sit out in boats waiting because the coral are getting too far apart because of the rising ocean temperature,
Right.
and then they wait for the confetti and scoop up all the confetti to try to help it drive their boat over and dump all their confetti over on the neighbor's yard.
Yeah.
That's
Right.
which
Right.
is neighbor's yard is a new euphemism I'm
Right. I
definitely
was
going
like,
to
yeah,
be using.
confetti in neighbors' yards is an interesting euphemism for like
A lot of
gametes.
things. Yeah.
Right.
After this podcast, my dirty talk is like going real next
Yeah.
level.
Well, I have to say it's funny. Like there's definitely been moments during my PhD where we've had in our lab heated arguments about collecting sperm and eggs,
Sure.
and speaking super loudly. And I remember once being like, if anyone walked by right now, they'd be like, "What is going on in there?" So.
Well, this is related to the other question that I definitely, I think it'll lead us in the right direction, and I don't want it. Why do I often like explain the reasons why I'm asking a question. I don't know if that makes it more interesting or less interesting, but that's just what I do. Here we go. So you were talking about taking kind of fossils, right? You were saying like you're kind of scraping. What was the thing?
They're not fossils.
They're not
just
fossils.
they were alive organisms, and then we put them in ethanol, which kills them, and then we look at them under the microscope.
Ah,
Yeah.
I see. Okay, never mind. Well,
But then one of the things I'm scraping is the like all the bacteria that eat the sulfur are usually growing on those same plates. So that's what I meant by the scraping.
I see. Well, I mean, I guess my question will still work. So I was thinking about the ways in which scientists try to deduct or use inductive reasoning or whatever to figure out more about species and life. And for example, you find some skeletal remains of some primate that's never been discovered before, and you see some way in which like the femur, like you have a little bit of a femur and you can see like some wear and tear in it in a certain way that and from that you can deduce that they were able to stand upright, or they were upright a fair amount of the time, or they have a bone in their finger that is bent more than humans, so maybe they were still climbing trees at the time, or you find some teeth and you realize that it was able to eat fruit, and from knowing that it ate fruit, then you know that it's probably a territorial species, because vegetation, you don't need to be as territorial, and fruit is more scarce. So mammals that need to be around fruit trees are often a lot more territorial. So you can figure out all of these other patterns from these little bits of evidence. You can find a whole bunch about what their entire lives are like. What kind of stuff? I want to ask that because I think listeners might be like, okay, this lady finds out why my house, how sea scallops blow their confetti. What is she just into sea scallop sperm? What's going on? But there's all of these really important lessons that we can learn from the tiniest little bits of evidence.
Right. And so I would say that the most important, or I shouldn't say the most important, but the most directly relevant, line you can draw between looking at, say, sea scallop reproduction, which I haven't even really gotten into yet, but
Oh, we will.
yeah, is how it impacts your ability to fish them, right? Your ability to eat them. So knowing how your food reproduces is pretty important. Otherwise,
Right.
it's very easy to kill it all, and then you don't have any more food, and or that food. And then it also has all these other impacts on the ecosystem, which might impact other things you care about, whether abstractly, culturally, deliciously. I don't know. It depends, right?
Yeah, it's true. We do need to eat. All right, I'm sold. I guess this is important.
Yeah. Yep. So it's also just interesting too.
Yeah.
And you know, there are things that you might be able to draw about other species after doing an in-depth research on one species that's similar. So presumably some of the information we might learn from sea scallop spawning could be helpful for other species that spawn in a similar way based on what little we know about them. So, you know, and one cool thing about science too is you never know, you know, 30 years from now, if that random paper you wrote that like 2 people have read is maybe useful to someone's project suddenly, because that does happen.
You gotta hope.
Right. You hope, right?
When you're writing it. You
hope.
Damn it, I hope someone's going to see this thing.
I have at least one paper that's a tome for my PhD. It's just so big, and I'm like, I don't know who has time to read this, but it's a great review paper on a very particular subset of issues if you're a grad student.
Well, no one's ever going to argue with that because no one's ever going to put the time in
to read it. Yeah.
So when you're studying sea scallop, do anyone ever eat their homework? Anyone ever have that excuse?
Well, I don't really. So I guess I'll explain more about what I did with sea scallops.
Sure.
Other than confetti. The main question that for my PhD work was whether or not population size or density of the scallops. So density is like how many individuals you have in a given area. And population size is just like the whole population size number of scallops. And so we want to see if they're really far apart. Do you get less eggs fertilized by sperm? We'd assume that if they're far apart, the chance would decrease because they're farther apart. And if they're close together, it would go up. But that relationship actually with sea scallops isn't super linear. When we put them in little nets, it was a very clear relationship. Didn't have a lot of scallops. They were far apart. Not many eggs got fertilized. Put a lot together in a small space. There were lots fertilized, right? Mm-hmm. But then when we put them out on their own, on like they're on the bottom, that we had stakes underwater, and then we would go and monitor them, and we would collect eggs from female scallops spawned in the lab, which is actually really hard because they spawn when they want to, so you kind of have to be there all the time. So I spent a lot of time sleeping under my desk and waiting for scallops to spawn. And so you put these like tiny, tiny little eggs, they're orange, in these containers that have mesh around them. And the mesh is so the sperm can swim in and fertilize the eggs, but the eggs can't get out. And so we take those from the lab, and we'd have to rush because the eggs were only good for 8 hours. And so we'd take them in a boat, drive down this estuary, and then put them down underwater on the bottom and hope that some males are spawning, and then collect them the next day and hope that some of the eggs got fertilized. So there's a lot of ifs and hopes, but we did get data. But what was really interesting is that the population where they were more spread apart had similar numbers of eggs fertilized as the one where they were all close together, which we thought was kind of weird. But the hypothesis that I have is that...
Can I take a whack at something? I want to be wrong first, and then you tell me the correct thing. So could there be some sort of like if-then biological mechanism where they're able to kind of sense when there's like when they don't have a lot of neighbors, there's something triggering them to be like, we better blow a lot of confetti, and then when things are getting tighter, there's maybe some benefit to
holding off.
Holding off a little bit. Some of the mating. It would be strange from like a selfish gene perspective to do something like that, because that almost sounds more like group selection or something. But I've had 30 seconds to think about this, and now let's find out how I'm wrong.
Well, right. So it would probably, like, the assumption is that it's the opposite, right? Because presumably all animals are wanting to increase their reproduction success, right? And they're not necessarily thinking about their self-esteem.
Right. Well, that's why I'm saying like thinking about it in a group selection way would probably be incorrect.
Right. So, I mean, it's not like we sit there on the bottom in scuba diving and just sit there and watch them. Mm-hmm. Although, you know, people do try to set up cameras and stuff to see behavior of things. It's really hard in like a natural environment when the water's not very clear. Because like these cells that they're releasing, the eggs, are like 70 microns across, and I think the width of your hair is like 100 microns, right? It varies, but it's something like 100 microns, so it's like less than that.
Mine's like 140. I got like 140 micron hair,
which is I think I was looking at the range. It was something like 50 to 150, because I was trying to explain to my class how tiny certain things were in hair widths, because I was like 200 microns.
That's how I measure all of life. That's and that's how we know if we like how close we got, and like, it's just by hair. I almost had all the best things happen to me. Missed it by a hair.
Yeah, I mean, like you could start measuring things in beard hair thread counts, right?
Yeah. I'm already on it.
Right. So the idea is that like if scallops are alone, they're not going to start spawning. Maybe if they're dying, that would be possible. But
how many dogs you got around there?
We have 2.
Woo! So, into it. All right.
Yeah,
sorry
that
for
one that
I
just
just
walked
got
by.
I got very excited for you. A lot
We
of
had a
people just do
cat,
one
too.
dog going.
Yeah.
I like the double dog situation. Pack of animals.
That one, the tan one, is 17 years old.
Oh my goodness.
Yeah, she's
that
very...
is a long, wonderful life.
Yes.
Looks pretty good.
Yeah, she's I don't know how that dog is still alive. She's done a lot of things that would kill most normal dogs. So
all right, sorry for my dog tangent.
No, it's okay.
I love your dogs.
One of the reasons why I don't have a Zoom background is because my students like seeing my dogs when they come by.
Yeah.
So it helps. I think. I hope. Oh yeah. So when there aren't many scallops around, we think that they might be swimming towards each other, because in some of the data we found, it looked like, because we would do surveys with divers, that I would send the divers out and they would look at how many scallops were in each area. And it looked like on some days that the scallops that were far apart had like started clustering themselves. So my hypothesis is that they might be doing that to increase like the percentage of eggs that get fertilized. I don't know for sure. I'd have to do some more experiments, and I was at the end of my PhD at that point. But that's what I think might be going on. So, and there's a lot of things.
You were our only hope. And now we'll never know.
There will be another poor grad student someday sent on a reproductive PhD. They're very difficult. Reproduction work is difficult, especially if it's related to fertilization, because there are very few animals, like marine animals, that are easy to get to spawn whenever you want them to, and a lot of the time there's a lot of waiting, and some don't spawn in captivity well. Sometimes they just die. There's so many variables that are really hard to control, and so there's sort of a limitation on questions you can ask with our current technology and know-how. So, oh, you know what? I didn't tell you the big thing about my adventures with scallop gonads.
You holding out on me?
Yeah, I always forget.
Well, spill it.
Well, so one of the things that I did is I measured gonad weight, or what percentage of the body weight is gonad. And so when sea scallops have a huge percentage of their body turn into gonad, that means they're about to spawn, right? Mm-hmm. And their body can be like 30%, 35. It's like, that's like both your legs. Imagine if both your legs became all just like
sperm.
gametes. Yeah, just sperm legs.
Just
sperm
walking
legs
around
all day.
I mean, depending on how long quarantine lasts,
right.
That might be a reality.
Right. So that's just to give you an idea of like how much their body turns.
That's amazing. That's also how much sloth poop.
Right.
Did you know that?
Is it? No, I didn't know that.
About 30% of their body weight. Well, it depends on the species, but yes.
Yeah. So that's sort of how
what would you rather? Be 30%
gamete? 30% poop. Yeah. I don't know. I feel like you should pull your listeners on that one.
Yeah, all right.
So at one point I was trying to figure out by doing these dissections, right, where you look at the percentage of weight. And that's also, I never really ate my work because when I did dissections, I get sperm and egg all over my hands. And so I didn't really want to eat it afterwards.
What about you're out to a fancy dinner? You eat scallops, or is that,
I got mixed feelings? I'm still recovering. I encourage other people to eat it, but it's just...
You encourage them to?
Yeah. I mean, if I think sea scallops as a species in general, most scallops you buy are probably harvested or grown sustainably. So.
Okay.
Sorry, I have the growl sisters behind me right now.
I love it.
Anyway, I was trying to figure out with these gonads, when they get bigger and smaller, if there was differences between where I was working in Maine and then farther down east at Mount Desert Island, where Acadia National Park is. And I started working with this fisherman, Andy. And Andy is a bit of a character, kind of wild. He's, I think interesting is the best way to put it, but he's a little bit all over the place. And I went to meet him at a
15% gamete, 15% feces, this guy.
Which guy? Andy? Oh, yeah.
He's a real mixed bag.
He is a mixed bag. It was. So he was meeting me at a gas station to give me these. I like
when I said that, how accurately confused you were when I made that super dumb joke. You had the exact correct response. We'll go with it. All right.
So, we're meeting at a gas station. He's been collecting these gonads all summer, and they're in these little things of formalin, these little containers, and it's this big white bucket with a cap on it. And I had given him instructions and all the formalin and stuff. And in Maine, no one locks their cars. Okay, like no one locks their cars there. Really, I mean, occasionally, but generally speaking, it's just not.
I'm so excited for this story. So
I'm waiting for him at one end of the parking lot, and I can see, because he has to drop his kid off at the doctor's, which is exactly across the street. And I actually remember I was sitting next to an old telephone booth, and I saw someone use it. And this was even 9 years ago. I was like totally fascinated. He had seen another car come in that had a University of Maine license plate on it, which is where I was at the time, and he assumed it was mine. And the car was unlocked, and the person had gone into the store, and so he just put the bucket in their car and drove across the street. And so I didn't see him till he drove across the street, and I was like, oh, well, where are my samples? He's like, well, I put them in your car. And I was like, well, it wasn't my car. And then we look, and the car is gone, right? Just gone.
Wow, that's amazing.
And so it gets more amazing.
Okay.
I call all these people at the university. No one knows where this car is, which is crazy because it's a university car. His wife posts on Facebook, calls the local paper, puts a wanted ad in the police station. A woman who was driving the car connects over Facebook. A bunch of news articles get written about it, including like Associated Press. And it was like, man loses guts. And then I wrote about it on my blog that no one reads. And then the next day, I got an email from a producer at the Colbert Report. And,
really?
Yeah. And so they came and did a piece. It's 7 years old now. You can watch it online. They made it like a murder mystery piece of the missing scallop gonads.
That is incredible.
Yeah. And
look at you.
Some would say that was my start in science communication. My thinking was when that happened, because no one knew who I was, and then it went pretty well. So I was like, well, if I didn't mess that up, then I must be doing something right. So
you're it's so weird that your life is okay. So you were studying scallop porn when you weren't scuba diving or in a submarine, but the newsworthy thing that happened was just someone put a bucket in the wrong wrong car.
I mean, it was hilarious, and if you watch the clip, you'll see why Andy is a gem.
Yeah.
But yeah, it was a big to do, and it was fun, and I'm still friends with the producer. She's pretty cool. She's moved on to other things, but she's great.
Nice.
Yeah, and
okay.
So yeah.
Well, I have because I don't want to take up your whole day here, but I have a couple other things that I wanted to ask you that are, I think I had 2 ridiculous questions. And did I have a good one? Jeez, I'm not sure that I had. This is what I wanted to make sure and ask you. One, what kind of a submarine are we talking about? I feel like if I was a listener to this show and the host didn't ask you about being on a submarine, they weren't doing their job. So it's not exactly sciencey, but
no, it's okay. I didn't know if you were going to tell me what both the questions were, or you want me to answer one and then the other.
I'm trying to remember what the other one was.
Okay. Well, so lucky for your listeners, I told a story about what it's like being an Alvin on the Story Collider.
I'll make sure and put links on
I can send you all my stories.
Yes.
There's a bunch of them on there, but I'll tell you that it's like a 6-foot diameter sphere that you're in with 2 other people. So it's tight in there, and they don't let you wear anything synthetic because of fire hazards. I guess you'll burn more slowly if you're wearing cotton or wool instead of anything synthetic. And it sort of looks like, I don't know, what you'd imagine the inside of a spaceship would look like, with all the lights and buttons and stuff all on the inside.
Can I pause for a second?
Yeah.
I was just taking in that. Can you just say all of that one more time?
Which part?
The thing about something like broke in my brain thinking about burning alive on a submarine.
Right. Well, yeah.
So
I'm just
and then I just my brain just shot off into all of these weird I just had an action movie go off in my head and stopped listening to you. Straight up, that's what happened.
Yeah, well, I'm thinking, I'm sort of going through some of the highlights of when they gave us the tour before we got to go in, and because they show you, they open up the top and you get to look down, and you're like, wow, that's a small space for 3 people, and there's this little ladder. And then there's like CO2 scrubbers, because it's a compressed the air it's sealed off, right? And so there's only so much air in there for 3 people for so many days. The idea is that you come back within 12 hours, but there's enough for, I think, 3 days in there. And so it's pressurized. So, there's concern. I don't know exactly how it works, but there's concern about fires, right? And so one of the things they said, they're like, you have to wear cotton or wool. So all the things I wore, none of it was supposed to be synthetic, because it burns more quickly. But actually, though, the most horrifying thing that they told us, they're like, if you get trapped on the ocean seafloor, 1.5 miles below the surface, I think that's how deep we went. You get trapped down there for like 3 days and no one can come get you. You see that lever down there, you turn it, and the metal sphere that you're all in, right? It's like the main compartment, but there's still like the rest of the sub attached to it, right?
Yeah.
Would be released from the rest of the sub, and you'd go shooting to the surface. And they said, well, we don't know how fast it would go because it's never happened. So hold on to something if that happens. And I was like,
you
great,
don't
I can't
test that.
Well, I don't know how you would, but yeah, I remember thinking, well, great, I can't wait to die that way. So
you can't put a sub with no one in it with the monitor thing and then have the thing lift off.
But their safety record, I think for Alvin at least, I can't speak for all submarines, like small submarines, but they have a really good record. And the engineers who work on it, I remember watching them on my trip. They basically put it together and take it apart, like the outer parts, every day to check. They check everything, every trip. They spend so much time cleaning it, taking care of it, replacing parts. Like they really know what they're doing. So yeah. I felt very confident, but it was kind of in the safety briefing of like, well, if the worst possible case scenario happens, this is what you do.
Yeah.
So
that's why I can't own a submarine. I just don't trust myself to keep up with the necessary maintenance.
Yeah, that's the only reason, right?
Yeah, it's the only. I mean, I've come close a lot of times.
Well, where would you put one if you had one?
In a trailer. I drive around with my.
Like, check out my submarine.
Yeah. Well, because I want to be able to go into a lot of different places with my submarine. I travel quite a bit, and that's well, then you really gotta not just to know how to do the maintenance, but then you're gonna get a lot of questions at gas stations too, and you're gonna the mechanics are gonna be very interested in the submarine. You're gonna have to know what you're talking about. They're gonna ask you if it has a Hemi and all that. So, I considered all these things and I thought, you know what, maybe a submarine isn't the most practical thing that I could buy right now.
No.
So we'll see. Maybe in the future. Someday.
We can dream.
Has anyone ever been stuck for more than 12 hours in Alvin?
I don't know of a story. That doesn't mean there isn't one that there might be one that exists, but I personally don't know of any.
When you were saying the scariest thing was, where my brain was that they were going to be like, if you get stuck on the bottom for 3 days, you pull this lever and it kills you.
No.
And I'm relieved that you get a chance to hopefully not get the bends and shoot to the surface.
Well, so it's all pressure, right? Like when they seal it off, supposedly all the pressure inside there is the same, and so the steel on the outside is like withstanding the pressure from the outside. That's my understanding of it. But someone who's an engineer,
it just might be a rough ride.
Right. But it's still
you don't got seatbelts in those things.
I don't remember any seatbelts, but. It was a long time ago. I went in 2007 when I went.
Okay, so
It was a long time ago. Maybe they have seatbelts
faint memory
now.
of seatbelts. All right, I have 47 more questions about submarines, and then I can let you go. No, I have that was mostly all that I wanted to ask you. I was still curious with the going back to the vent, and you have this whole ecosystem around a vent.
Mm-hmm.
Vent goes away. Has an event like that happened where it's just like, time's up? We happen to be there at the sliver of time when this event that's been around for 1 million years ends, and now this whole little ecosystem is destroyed.
Yeah,
I mean,
well,
that must be a thing, right?
It depends on the vent system. The vent system that I got to visit, they had eruptions every like couple of years to a decade. It would, so it wasn't an uncommon event. So I think the animals were kind of adapted to having what seemed like a lot of larvae in the water column too. And so they were able to colonize new vents that emerge, because sometimes, there's a lava eruption and it erases one, but then other ones might pop up.
That's amazing. I mean, these things are you're already just blasting confetti into the void, hoping for something to survive. But you also, as an individual, your entire home goes away, and you look around at the vast, infinite, whirling chaos around you, and you just go, I guess we'll head that way, maybe see if we run into something. Do they have like ways of
the scale of these animals and the scale of the eruption is probably, I don't know, but I'm assuming that the lava for a lot of it just like covers them, burns them into oblivion. A lot of the animals I studied tend to not move very fast, and or were attached to the bottom. But I could see maybe some fish or crabs.
Oh, I remembered my other really stupid, ridiculous question. I'm sorry to, whatever. We've had a lovely, why am I apologizing for having fun on my own show? You're having fun. We're both having fun. I'm apologizing because you may not have good answers for this. Do you know anything about the history of sea scallops, or humans' use of sea scallops? I was just thinking about how I don't really care for sea scallops, and then it got me thinking, who put the first one in their mouth, and when did they become popular? And just on the off chance that you happened to dig into some sea scallop fun facts that had nothing to do with your scientific work, I thought I would ask.
Yeah, so those are good questions. In terms of
generous.
No, let me. Scallops for most people are incredibly delicious. I do think that.
Oh, yeah, there's a lot of people that are wrong about a lot of things.
Right. I take it you're not a seafood person.
I'm not a huge seafood person, and I'm being silly. I'm fine with scallops. Hold the calamari, like shrimp, oddly enough.
Yeah. Well, my favorite seafood is crab, but I really like working with bivalves because I think they're really cool animals, and I also like working with the people in those industries. So there's multiple reasons to like an animal, right? Mm-hmm. But yeah, so people have been eating not just sea scallops, but the bivalves, anything with the two hard shells, right? They kind of look like rock food mm-hmm. for a really long time, like thousands and thousands of years. And there's like hundreds of scallop species all over the world. And scallops have been like, maybe not sea scallops, the species I worked with specifically, but there are a lot of scallops that have been used like symbolically in cultures too. Like there's a pilgrimage in Spain.
I'm so happy I asked this.
Right, and I'm forgetting the name of the saint. I want to say it's like St. John or something, but there's this long pilgrimage. I would have to Google it. That the symbol of the saint is the scallop shell, and he apparently would like go door to door with a scallop shell and ask for just enough food to fill the shell. And so that's my understanding of why it's the symbol for it. And it's also like a fertility pilgrimage. So some people who do it because they want to get pregnant. So there's symbols of fertility, which to me is something I connect with quite a bit because I worked with gonads so much. And then, think of like, I'm going sort of on a cultural rant here, but like the Botticelli painting of Venus coming out of the shell. Right? She's the goddess of love, and she's on a giant, giant shell in that painting. But it's also a symbol of, I think I was reading in I don't know if it's Irish culture, but it's also like the symbol of like the setting sun. So it can mean death too in certain cultures. So there's a lot of cultural significance, and the shells, have been used on all sorts of like emblems of family crests and whatever. So there's a lot of really interesting history of its symbolism. And so presumably people have been eating them for a long time. And I think there are divers that dive for them in like, I want to say Japan, but there are some other countries as well with cultures have been doing diving specifically for scallop species and other species, but for like a really, really long time. I don't know if it's hundreds or thousands of years, but so there's a lot of history, and lots of history we don't even know about. And I could go on about like oyster shell middens and I, in terms of like weird things that people eat from the sea. I think the weirdest thing I just have a hard time wrapping my mind around is, have you heard of hackarl? I don't know if I'm saying that right.
No, but I'm excited.
So, Greenland shark. Have you heard of them?
No.
Okay, they're in like northern seas, at least the North Atlantic. They can be really old. I think there's some estimates that there's like found one that was like 300 to 500 years old. I don't know. I'm not a shark person. They're really old, and if you were to eat them raw, they're like filled with ammonia, which is poisonous. So someone, I don't know when, in Iceland took presumably a dead Greenland shark. I don't know if they killed it or it washed ashore, buried it for some long period of time.
Yeah.
Like the ammonia, I think, comes out of it or whatever. It's sort of like a curing process. And then they dug it up again and ate it and didn't die. And they're like, we're going to keep doing this.
That's how we do it. Yeah. Yeah. So, if at first you don't succeed.
So, and I don't know if
bury your food, dig it up later, and try it again.
Right. And I don't know if I've got like all the poisonous compounds. I know ammonia is a big part of it, but I don't.
We'll get the fact check team on it.
Yeah. But when my husband and I went to Iceland, there's a Greenland museum. Greenland Shark Museum, and you can go and see their museum, and it's very small.
And they have samples.
Eat it. Yeah. Yes, they have samples. I did not eat it, but my husband did, and he thought it was okay.
That's a lot of work for okay. That's a big risk for okay. That's a bit of a sunk cost fallacy situation with once you've like buried the shark, you did the thing, you survived, then you got to tell yourself a narrative that like, yum.
Right.
Look at this novel thing that I figured out.
Now they hang them that place. They hang them in sheds, these slabs of meat. Just hanging out there.
Yeah.
They don't. I don't think they bury them anymore.
Yeah, I like that move. I thought the burying process was okay. All right. Well, I would try it. I'm not gonna lie to you. I would absolutely try it. All right. Well, I have to tell you, my sea scallop, my sea scallop shell overfloweth with information. You
have one behind you, don't you? What is that painting behind you? The blue one. Higher up. What is that?
Some sort of I guess it's like some sort of trippy oyster shell.
But
thing.
That's like more of a scallop shaped shell.
Is it?
Yeah.
This is
that looks more like a scallop in a lot of.
Zachary
it looks like,
Brown, I think, is this guy's name. I get a bunch of art from people that make cool things. I gotta change this one out, actually. There's been a bunch of... but this is what a sea scallop shell looks like?
Some look similar to that.
Does it have a weird maze in the middle, a labyrinth?
Some, yeah, very special ones.
All right, so where can people find everything that you do other than the links on YouTube and my site?
Oh man.
Just
I do have a skylarbayer.wordpress.com site, which I try to update with links to all the things I've done, including my research. And I'm hoping to develop a better page as now I'm a faculty member, but that's where you can find a lot of what I've done. And I'm on Twitter. So at Dr. S.R. Bayer.
And can you fix my Wikipedia page for me? What happened like 8 years ago or something like that? Some dude was like, hey, big fan, I want to write your Wikipedia page. I was like, have at it. I mean, who am I to stop you? I don't have an understanding of how that works. Go for it. And it's, he must have got a collection of, bless his heart, but he must have got a collection of newspaper articles which bias towards like creating these like very dramatic like narratives of like, and this comic that started out on an open mic rose to the level of a this and that, and it's like, so, and that's sort of how my Wikipedia page reads, because it's all based off of these like silly newspaper articles that don't usually put stand-up comedy in their newspapers. So when they do, they try to make it seem like a real hero's journey. And so it's embarrassing. And I want to update my Wikipedia page. So when we're done recording, I'm going to ask you 2 brief questions about that. But thank you so much for joining me. And
yeah, well, thanks for having me, Shane.
Doing Story Collider and all the awesome science communication that you do. Good luck with COVID teaching and teaching remotely. You might find that teaching remotely is a cool fit for you. I think some people are taking to it pretty well. I think that I've talked with some professors that are like, I kind of like it, but
yeah,
there's pros and cons.
A lot of my work, a lot of my strengths have been in-person interactions. So it's not, I suppose it could be worse, but I personally would prefer to be in person again someday, but safely.
Yeah. Well, 2 years. That's my estimate. Yeah. Awesome. Well, thank you so much, Skylar.
Oh, thanks, Shane.
And thank you, listeners, for being such wonderful, curious people.